Dear students, so today the topic to be discussed is AIDS, means acquired immune deficiency syndrome. This particular topic belongs to the course Medical Virology and Parasitology, Module 3. So this is the last topic of Module 3. So before going to start the main topic, the detail, the question should be in our mind that what is AIDS? People have learned uh, about this particular terminology. So the full form of AIDS is acquired immune deficiency syndrome. All the words have some meaning and as a whole is actually exemplifying the particular syndrome. So here acquired means something that you are gaining, means to come into possession of something new. Next is immune deficiency, means it is decrease or weakening of the body's ability to fight against infections or any kind of illness. Then syndrome is not a particular type of disease. It is a group of signs and symptoms that occur together and characterized by a particular abnormality. So that is known as syndrome. So human immune deficiency virus, actually its infection leads to a weakened immune system, it's an acquired immune system, if not innate immune system, weakening of the acquired immune system. And this particular infection makes a person with HIV vulnerable to a group of illness, that is opportunistic infections that would not as easily affect a healthy individual. So first, this particular type of virus was identified in 1981 in USA among the homosexuals. So in 1983, French investigators named this particular virus as lymphadenopathy associated virus, LAV. Next year, the virus was isolated by Gallo and his co-workers from National Institute of Health in the United States. And they named this particular virus as human T-cell lymphotropic virus type 2, sorry, type 3, HTLV3. And in 1986, a new strain of HIV was isolated from West African patient uh, with AIDS, which is called HIV-2. And in that very year, ICTV, International Committee on Taxonomy of Viruses, gave a new name to this particular virus, and henceforth it has been regarded as or called as human immune deficiency virus or HIV. Since its identification, HIV is indeed a devastating disease of the mankind. Now, what is HIV? So, human immunodeficiency virus, that is actually infecting the human being, not the animals, only the human, decreasing or weakening the body's ability to fight against a particular type of infection or illness. And this is an obligate parasite that needs a host to replicate and proliferate. This particular virus belongs to a group of retroviruses called lentiviruses. And they have the genome, it is RNA, along with the two RNA single-stranded chain that contains a special type of enzyme that is called reverse transcriptase, and that is why they are called retroviruses. This particular virus needs a host cell in fact, and the RNA, that is the genome of the virus, first transcribed from viral RNA to viral DNA, which is done by the enzyme known as reverse transcriptase. There are two types of HIV. One is HIV-1, and second one is HIV-2. The virus should not be here. This virus should not be there. So HIV-1 is the most common type of HIV, and mostly you can see in sub-Saharan Africa and throughout the world you can get this particular type of virus, and human can get infected with this type of virus. The groups are MN and O. A pandemic dominated by the group M, and it has some subgroups A to J. HIV-2, it's most often found in West Central Africa and parts of Europe and India. So as per CDC, Centers for Disease Control and Prevention, HIV can be classified into a different mode. So their perception is that according to CDC, disease staging and assess the severity of HIV disease by CD4 cells counts and by the presence of specific HIV related conditions. They have classified HIV into three different categories: A, B, and C. Patients in categories A3, B3, C1 to C3 are considered to have AIDS. Now, what is actually the morphology of HIV? It's an envelope virus. The outer coat of the virus is composed of bilayer lipid. Different proteins are embedded in this envelope, viral envelope, and actually they are forming spikes consisting of outer glycoprotein and some transmembrane proteins. The lipid membrane is actually they are getting from the host cell cytoplasm during the budding process. When they are coming out from the host cell, they are getting this envelope, or they are borrowing this layer, external layer. Below this envelope, HIV has a matrix protein, and this particular protein lies between envelope and the core. Viral core contains viral capsid and nucleic acid, two single-stranded RNA, along with an enzyme that is a HIV-specific replication-specific enzyme known as reverse transcriptase. So their nucleocapsid contains reverse transcriptase as a vital enzyme. Along with that, they have another three enzymes. These are protease, ribonuclease, and integrase. So integrase is very much essential for integration of the virus DNA to the host DNA, means provirus formation. So this is the overall structure of HIV. Here you can see this is the core part. Here you can see this is the core part, and it has two single-stranded RNA, and now it has reverse transcriptase. It has some other enzymes here, and this is actually the capsid, and as a whole it is known as a nucleocapsid. So this particular capsid is composed that they have some enzymes. This core has some particular type of protein that is called the capsule protein, It is P24 protein, and this capsule protein is very much specific and is very much important for the identification of or laboratory diagnosis purposes of this particular type of virus. So core, and this one is the outer layer of the virus. It is nearly spherical. So this is the outer surface, and this outer surface is a bilayer lipid membrane. And this bilayer lipid membrane, they derive this lipid membrane that is known as the envelope of the virus from the cytoplasmic membrane of the animal host. And they have some spikes. Here you can see these are the spikes. These are glycoprotein and transmembrane protein. Here you can see these are the outer protein and the transmembrane protein. The red one is the transmembrane protein. So they have the two type of glycoprotein, and this one, the red one, is known as GP41, is the envelope protein, and the violet one here is violet one. This is GP120, glycoprotein 120. This is also a particular type of envelope protein. And in between these two, this is the envelope, and this is the core. So in between, they have the matrix. This yellow dot here, this yellow circle you can see. This is the matrix protein, and this matrix protein, this matrix protein is actually known as P17 matrix protein. Here, this matrix protein is P17 matrix protein. So here you can see four different type of protein. This is the capsule protein, P24. This is P17, this is the matrix protein. GP120 is the envelope protein, and GP41, this is another envelope protein. So in the different it has some different in structure. From the previous picture, you can see that it has some different in structure from the other retroviruses, and it has a diameter of 120 nanometer. It's quite large and roughly spherical in shape. Glycoproteins are there, the spikes protein. They are actually responsible for or needed to attach the virus particle to the host cell, and the transmembrane is critical for cell fusion process. So to make an entry to the host cell, these transmembrane proteins are very much essential or critical. HIV-1, the first type of HIV virus, HIV, actually human immunodeficiency virus, is composed of two copies of non-covalently linked unspliced positive sense RNA molecule, single-stranded RNA molecule, that are enclosed by a conical set composed by the viral protein. So you have already seen this one. This is a nearly conical set. Here the capsid, nearly conical set. So conical set capsid composed of the viral protein. The RNA component is actually 9749 nucleotide long, so near about 10 kb long, 10 kilobases long. Now HIV genome, so RNA, single-stranded RNA, two single-stranded RNAs are there, and they consist three major proteins. These are three major indispensable viral proteins, and that proteins are actually, they have some genes. So three major genes are there that are encoding different proteins, that may be structural protein, maybe functional protein. And these three genes are known as GAG, POL, and ENV. GAG gene is actually encoding for group-specific antigens, capsid protein, then POL gene for enzyme polymerase, protease, and integrase, and ENV gene is for envelope-like protein. So here you can see both GAG and ENV, they are actually synthesizing group-specific antigen, capsid protein, and envelope protein, glycoprotein, not envelope, glycoprotein. And the POL gene is actually responsible for a synthesis of the enzymes like protease, polymerase, and integrase. The proteolytic cleavage of the polyprotein encoded by the ENV gene leads to production of viral glycoprotein. So here we can see that we have already seen the GP41 and GP120, but first the polyprotein that has been synthesized by ENV gene is actually GP160. The GP160 latter is cleaved by the protease, viral protease, into two subunits, that is GP41 and GP120 that you have seen in the envelope. One is a extracellular part, another one is a transmembrane. And then glycoprotein 62, that is HTLV1 specific, cleaved into two another protein subunit that are known as GP46 and GP21. So this is the HIV genome and how there is post-translational modification going on in HIV. So testing for the viral infection and how our body responds, because it is actually decreasing or compromising our acquired immune system. So definitely infection of HIV will affect the immune system of the host. So this can be utilized for assessing the viral infection or for testing viral infection. So here in the left hand column you can see few bars, a few days are mentioned, day 0, 7 to 14, 16 and so on. So day 16 and day 22 is very much important, because day 16 in the blood of the patient you can check P24 antigen. On an average 16 days, 95% people develop P24 antigen within 1 to 8 weeks. Then 28 days, after 4 weeks, 95% people will get will develop antibody by IgA and IgG. So after 28 days or 4 weeks. And more than 99.97% people develop HIV antibodies with 3 months of time. So how it is actually invading, it is actually creating problem or infection in human cell. So the pathogenesis of the particular type of virus. So HIV actually infect the CD4 cells, then fuse with the host cell, attaches via receptor, surface receptor, and then fuse with the host cell. Then within the host cell cytoplasm, uncoating took place, releasing the viral RNA along with reverse transcription tryptase. So then they will go for reverse transcription, means from viral single-stranded RNA to RNA-DNA hybrid and DNA, and this particular DNA is viral DNA, and this particular viral DNA will be transported to the nucleus and will bind with the host cell chromosome or host cell DNA by the utilization of the enzyme integrase. This particular phase is known as provirus. So the proviral DNA will be there in latency, maybe for few months, maybe for more days or less. But when they will come out and will synthesize new viral RNA by transcription as well as protein synthesis, and then polyproteins are cleaved by the viral enzymes, proteases, viral proteases, and will eventually assemble to form a new variant and come out from the host cell by the process known as budding. In between that, this particular virus will create some cytopathic effect into the host cell. And these cytopathic effects should be studied to identify whether the patient is HIV positive or not. So this is all about the HIV infection that we have already discussed. The first binding with the CD receptor. Here you can see this is the CD receptor. First the virus binding with the CD4 receptor, CD receptor. And the receptor is CCR5, chemokine receptor 5 or CXCR4. So here you can see that these are binding with the AP41 of the viral protein, spike protein, and then CD4 positive cells with CCR5 or CXCR4 receptor. Then it is coming here, uncoating there, and then go for reverse transcription, and then we'll integrate, then we'll integrate here by the enzyme integrase. Then it will go for latent provirus. It may come out as a virus DNA. Then the nucleocapsid will be formed, and they will be released from the host cell, animal cell, infected animal cell. By the process known as budding, during that process, they will get envelope, or they will synthesize envelope and spike protein, and then go out, or come out from the host cell. Now, what are the events that happen in an HIV infection? So, in case of primary infection, here you can see this is a CD4 cell primary infection. Establishment of infection in the gut. Then you can see massive viremia, means presence of virus particle in blood, and wide dissemination to lymphoid organ. Then HIV specific immune response, so trapping the virus and establishment of chronic and persistent infection can occur. After that, immune activation mediated by the cytokines and HIV envelope mediated aberrant cell signaling. Then virus replication may be accelerated. Rapid CD4 plus T cells turnover you can see. So here you can see destruction of the immune system, mostly the acquired immune system, by the proliferation of this particular type of virus. So this is these are the events that can happen in our body after HIV infection. Now, host will respond to HIV infection first. In the acute phase, the reactant is known as the eclipse phase, where the host cell will not function properly. So onset of cytokines will come, and here you can see that pre-GP41 specific non-neutralizing IgM antibodies produce antibody virus immune complex will interact with each other, and CD8 T cells respond to the original virus. Then virus escape from CD8 positive T cells to new plasma mutant, and then autologous virus specific neutralizing antibody plus, and you can see this virus escape from autologous virus specific neutralizing antibody. So this is how the cell, how our body will respond to HIV infection. And next one is a source. So virus has been found mostly in blood, in semen, in cerebrospinal fluid, in lower concentration in tears, saliva, breast milk, urine, cervical or vaginal secretion. And to date, only blood and semen have been conclusively shown to transmit the virus. Next one is the symptoms. What are the signs of HIV infected person or AIDS person with AIDS disease? So there are two types of symptoms. One is a major symptom, another one is minor symptom. The major symptoms are weight loss, at least 10% body weight, then chronic diarrhea, at least for more than 1 month of duration, and then prolonged fever, same, at least more than 1 month. Then it can be suggested for checking HIV positive and negative. Also, you can get some minor symptoms or minor sign, that is recurrent oral pharyngeal candidiasis, then persistent generalized lymphadenopathy, and persistent cough for more than 1 month. So what is the mode of transmission? How this particular type of virus transmit? This particular, as I already mentioned, that this particular virus is a body contact. So mostly unprotected sex. So transmission from male to female is more than from female to male. It is nearly twice. To infected blood. So during sharing needles, use of contaminated needles and syringes should be prohibited. Then transfusion of human infected blood to blood products, it may happen. And next one, from mother to child during pregnancy, during childbirth, or during breastfeeding. Although in breastfeeding it is very low concentration, it's very low. Still, uh, we have to take care of that. From mother to child, this virus can also be transmitted. Now, what is the incubation period? The incubation period is not fixed. In few patients, it is for few months. In few patients, it is for few years, maybe 10 years or more. So it's not a fixed one. It's variable or uncertain. And what are the phases after infection with HIV? There are three different type of phases. Primary phases, phase 1, middle phases, a chronic phase, that is phase 2, and the phase 3, that is nearly the death phase. So in phase 1, it lasts 3 to 12 weeks. Acute HIV syndromes can be noticed: sore throat, fever, skin rash, meningitis, etc. Next one is the middle chronic phase, and this phase can last for 10 to 12 years. It's very competition between the virus and the host immune response is going on. That is why it lasts for 10 to 12 years. So patients are mostly asymptomatic, or some are showing mild symptoms, and here viremia is moderate. But in the third phase, is the full-blown of AIDS or HIV virus infection, and you can see severe immune depletion, the immune suppression for that. Severe immune suppression for that. Drop in the CD4 count, you can check, and also you can be noticed high viremia, means high amount of virus particle present in the blood. Now, this infection in a human can be staged into 4 stages. The first stage, that is the asymptomatic phase or asymptomatic stage, and this is the acute phase. Here you can see this is the acute phase. The blue line, that is actually showing the CD4 cell count, and the brown line, it is actually showing RNA copies or RNA copy number, that is the presence of virus particle in blood or serum, sorry, plasma. So this is the acute phase, where you can see this blue line means the CD4 cell count is gradually decreasing, and then here this virus RNA count, HIV RNA copies, is gradually increasing. But after a certain time, it is reverse. So CD4 cells counts are increasing, and HIV RNA copies per ml in plasma is decreasing. Then there is a second stage, that is called the minor symptoms. You can see some minor symptoms, as I have mentioned the minor symptoms there. So next one is a stage 3, that is the moderate symptoms. You can see the moderate symptoms, that is already mentioned, these modern symptoms. Here you can see CD4 T cell count and viral load. CD4 T cell count is constantly decreasing, but the viral load is constantly increasing. And the fourth stage, that is actually the last stage, or the death phase, the death stage of AIDS infection. Here you can see the CD4 cell count is very negligibly small, nearly 10-20, whereas HIV RNA copies are very, very large. Now, here you can see how to go for diagnosis. In case of laboratory diagnosis, there are 2 types of tests. One is a direct test, the second one is an indirect test. In the direct test, you can go for directly to check the antibodies. And for that, you can go with direct ELISA. enzyme linked immunosorbent assay, that is a serological assay. And you can go for virus isolation in culture, you can go for PCR, you can go for recombinant DNA techniques. In indirect text assay, you can check the CD4 cell count, you can check P24 antigen whether present or not, and lymphopenia and lymph node biopsy. You can go for this indirect test for laboratory diagnosis of HIV. Next one is prevention. So first thing is that raising awareness. It is crucial and essential. So what type of awareness? The partner, you must be faithful to your partner, and during sexual intercourse, you should use condom. That is actually prohibiting yourself, both the partners, getting infected by HIV, and not share any needless syringe. Distribution of IEC, that is information, education, and communication material to colleges, universities, and also throughout the areas, it is must. Advertisement from different media and channel, that may be television or any other means, is crucial for prevention of this particular type of disease. Next is the treatment. So there are different type of antiretroviral drugs that are already in market, that are in clinical trial. So this anti-HIV drugs, there are 5 different target points. They can attack these targets. What are the targets? First one is the chemokine receptor 5. Means if the target is receptor, means they are preventing the entry of the virus or attachment of the virus particle with the host cell. So this particular, if they are blocking this particular type of receptor, means the anchorage of the surface protein that is actually receiving the virus particle via GP41, it is hindering or inhibiting the interaction between the two, means host cell surface receptor and the viral glycoprotein. So the virus particle will not absorb, will not interact, or will not enter into the host cell. The second one is a fusion. The second, that is the transmembrane proteins that are actually responsible for fusion between the virus particle membrane and the cellular membrane. So if we can generate, we have already devised synthesized few inhibitors. So they are actually preventing the CD4 cell to fuse with GP120 or transmembrane glycoprotein of the virus particle, and inhibiting the virus to enter into the host cell, or inhibiting the virus RNA to come out into the host cell. Next is HIV reverse transcriptase inhibitor. So it is actually inhibiting the function of reverse transcriptase. We will discuss elaborately function of the in subsequent slides. So it is actually inhibiting the function of reverse transcriptase so that the viral RNA will not transform into viral DNA, and then if there is no viral DNA formation, so viral DNA will not integrate to the host DNA. Hence, there will be no proviral form. Next one is against integrase. So integrase is the enzyme that is actually integrating the proviral DNA to the host DNA. So function of integrase will be lost, means function, this integration will not be there. And the fifth one is HIV protease that is actually hindering the function of the polyprotein, means it is actually restricting the proteases to digest the polyprotein into subsequent glycoproteins, means from GP160 we can get 2 different type of glycoprotein, GP41 and GP120. So these are the 2 essential glycoproteins that are actually responsible for attachment and fusion of the virus particle with the host cell surface receptor via the host cell surface receptor. So these are the 5 checkpoints, these are the 5 target points that we can attack by using some either by drugs, or that may be synthetic, that may be naturally occurred. So this picture will tell you about the drugs and how we can we can inhibit the virus particle to enter into the host cell. So here you can see these are the inhibitors, that is Maraviroc, MRC, and uh, and Enfuvitide, ENF. They are actually the functional entry inhibitors. So they are interacting with the virus. This is the CD4 cell. This is the virus particle, and this is the CD4 not receptor is there. So these are the CCR5 receptor. So they are actually inhibiting this. So inhibiting entry. Next one is some drug molecules. They are mostly the nucleotide analogs. Uh, so this particular drug like zidovudine, AZD, lamivudine, etc., they are actually inhibiting the function of reverse transcriptase. And then the third one is the integrase inhibitor, raltegravir, RGV. They are active. The drugs that is actually functioning as integrase inhibitor. Next is coming out, maturation. It's a protease inhibitor. So the protease inhibitors, there are different type of protease inhibitors. You can see here, saquinavir, ritonavir, indinavir, etc., etc. So they are actually inhibiting the proteases, and then the polyprotein will not disintegrate into two subunits, or formation of glycoprotein will not possible here. So this is a particular type of drug, one of the drugs we have already mentioned. Zidovudine, or AZD, belongs to the family nucleoside analog reverse transcriptase inhibitor, NRTIs. So what is their actual function? Structurally, they are related to the endogenous nucleotides, thymidine. But instead of a 3' hydroxyl group, they have 3' azido group. So here you can see, instead of in case of the normal thymine, so here you can see they have OH group. So here in this position they have OH group. So this 3' OH group is responsible for this elongation of the chain, but instead of 3' group, instead of OH, here is the azide group N3 is there. So when this particular type of nucleotide analog will go for competitive inhibition with thymidine and bind with reverse transcriptase. So what will happen there? If this particular molecule, zidovudine, will bind with the reverse transcriptase, and it will bind with the nascent RNA or DNA, then you can see that no more chain elongation will take place. So this molecule is actually functioning as chain terminator. So no more chain elongation means no more DNA production, no more DNA formation. So no more integration and no more proliferation of the virus, or no more replication rather of the virus particle. So this is how zidovudine will function. Here you can see this is via the phosphate formation. First, this is the zidovudine. Via passive diffusion, it will enter into the host cell, virus infected host cell. Now, first by utilizing the virus thymidine kinase, this zidovudine will convert into their monophosphate. Then by utilization of the host cell kinase, they will subsequently transform into their diphosphate and triphosphate. Now this triphosphate will function as a nucleoside analog and will go for competitive inhibition along with the natural or normal nucleotide. And when this bind with the reverse transcriptase, it will just inhibit chain elongation or initiate chain termination. So that is how they can inhibit this particular viral growth. Thank you.